All right, welcome everybody. I'm Jenna Monroy, and together with Kiisa Nishikawa and Greg Sawicki, we're going to talk about classic and novel isolated muscle experiments. And so I'm just going to give an introduction to isolated muscle experiments and introduce the work loop experiment in particular. Then Kisa's going to talk about muscle avatar in her lab, and then last Greg is going to tell us about real world loads and unconstrained work loops. I just want to point out that we have a QR code for everybody to give feedback on our workshop today. I think Greg will post it in his presentation at the end. So I would just want to remind everybody to give us some feedback. All right, so where to begin? So I thought the best place to start is to remind us that muscles are highly versatile. They can perform so many different functions. And not only do we see that muscles perform different functions across different organisms, different muscle types, but even a single muscle can perform several different functions depending on the conditions. And this is why it makes it really difficult to predict muscle function during natural dynamic movements. And it's also why our main focus at IMSI is to understand muscle function across numerous different scales. So from the level of individual muscle molecules inside the sarcomere, all the way up through whole whole ecosystems and populations. And isolated muscle experiments fall somewhere in between at this tissue and organ level. And the advantage of isolated muscle experiments is that we can measure intrinsic muscle properties under different conditions. So we remove the organism and the environment from the experiment. And we can make predictions about not only the underlying molecular mechanisms of those particular properties, but we also can move in the other direction and make predictions about muscle function during natural movements. And the greatest advantage of these experiments is that we have a lot of control. And so when I think of isolated muscle experiments, I think of whole muscle experiments. But as I started to think about this talk, I realized there's a lot of variations. In fact, because muscles are so highly organized, we can measure properties from individual sarcomeres, like the picture on the top left, from a myofibril or strings of sarcomeres, a single fiber preparation, a whole muscle, or even an in situ preparation where the muscle is still somewhat intact in the animal, where they might still have blood supply and even a nerve for stimuli. And the type of variation that you might use might depend on your question. So if you were asking questions about molecular mechanisms, you might do a single sarcomere, myofibril, or fiber prep. But if you're interested in whole muscle function, you might be more interested in a whole muscle or in situ preparation. Now, in my lab, I primarily do whole muscle preparations where we take the muscle out of the animal and put it into a bath. And just to remind you that a whole muscle is an organ. So it's composed of both muscle tissue and connective tissues in and around the muscle, and it's connected to a tendon. Now, again, for the types of questions I ask, we try to remove the tendon and tie it off, but you could even do this type of preparation with the muscle and tendon intact and have the entire muscle tendon unit. And Greg might tell us more about that today. We will stimulate the muscle through the saline bath. And so in this diagram to the right, you can see these two parallel platinum electrodes where it would inject current into the bath to stimulate the muscle. And then we use a servo motor to control and record the length and force of the muscle. Certainly, as I mentioned, the strengths of these preparations is that we can control how the muscle is being activated. We can control the length and record the force, or we can control the force and measure the length of the muscle. The greatest weakness, and I'll say this several times today, is that it doesn't always reflect what's actually happening in the muscle during in vivo movements. So when I think of classic isolated muscle experiments, I think of the force length and the force velocity relationships. And so just to remind you, how to perform these types of experiments is that you can put the muscle in the bath and you would fix the muscle at a particular length. And then we would stimulate the muscle at maximally to obtain an isometric tetanus. And so at any given length, you could measure the muscle's passive force, that is the force of the muscle when it's not being activated, its peak force, and the difference between those would give us the active force of the muscle. And so we could fix the muscle at a particular length, and we can obtain the active and passive forces at each of those different muscle lengths and stretch the muscle out until we get a relationship of the active force and the passive force with length. And so to remind you that at short muscle lengths, the muscle active force is relatively low, it reaches a peak, and then it'll start to decline again at longer lengths, whereas passive force increases exponentially with muscle length. And so from these data, we can obtain the optimal length of the muscle, which is the length at which the muscle generates maximum force. It corresponds with the greatest amount of overlap between our thick and thin filaments and the greatest number of cross bridges between our actin and myosin filaments. Certainly, the benefits of this type of experiment is that we can compare across numerous different organisms and muscles, and we can make predictions about muscle force. So we might say that predict that a muscle functions at or near its optimal length in order to maximize its muscle force. But the limitations are that the muscles are pretty static. They are at a fixed length. They're not changing length at all, and they're maximally activated. And we know that this is not what's happening in vivo. So we have to take these things into consideration when looking at these data. The same is true for our force velocity relationship. So in this case, we're controlling the load on the muscle, and we're going to measure the force and velocity of shortening. So this diagram shows that we can give the muscle a particular load, and then in the middle, we can measure the constant force and the constant velocity against that load. And we can do this at various different loads to look at the relationship. And so on the far right, we can see the force of the muscle or tension as a fraction of its maximum on the x-axis, and the shortening velocity, that is muscle lengths per second, on the y-axis. And any one of these lines represents a different muscle, so that at really low loads, the muscle shortens really rapidly, and that the shortening velocity decreases as the load increases until it can no longer move that load. And so again, the advantages of this is that we can see that different muscles have different shortening velocities. So we can compare across several different organisms and muscles, and we can make predictions about shortening velocity and the power output of the muscle. But once again, the limitation is that these experiments are highly controlled. The muscles are under constant force and velocity, and that doesn't necessarily represent what the muscles are doing in vivo. And so looking at these data, it really begs the question of, well, how can we measure muscle function during dynamic movements? We know that muscles typically undergo continuous stretch shortening cycles during natural locomotion and other movements. We know that muscles can experience unexpected perturbations, and that force is going to change with both length and activation. And so it's been over the last 20 or 30 years that the work loop technique has gained some traction. So in the work loop technique, what we're doing here is we're going to control the length of the muscle, and we're going to subject the muscle to cyclical length changes, and we're going to measure the force output. And so what this little animation is showing is the change in muscle length is the blue line in the middle. And so we give them sinusoidal length changes. And we're going to stimulate the muscle at the onset of shortening. So we're looking at the force output of the muscle in red. And then we're going to plot force by length to get a work loop. And so you can read this work loop where the red dot indicates the onset of activation, and the muscle then shortens, and then it's stretched passively back to its original length. So the network of the muscle is the area inside the curve. And so again, the benefits of this type of experiment is that it's a better representation of in vivo muscle function, and we have lots of control. We can control the magnitude of the length changes, the cycle frequency of the rate of change, and we can change the phase of activation, that is, when in the cycle is the muscle being activated. But there are still some limitations. Here, muscles are typically being maximally activated, and they undergo sinusoidal length changes, which once again may not represent what the muscle's doing in vivo. Now, I think it's important to point out that muscle function depends on the phase of activation. And so just to point this out even further, this diagram is showing the sinusoidal length changes, and it's showing that when in the cycle, the muscle is being activated, dramatically changes the force and the work output of the muscle. So when the muscle is being activated during shortening, it functions like a motor and produces positive work, which is sort of this large orange work loop. Whereas when it's activated during lengthening, we see that the muscle functions more like a brake, and it's undergoing negative work. And then we can see lots of variations in between. So we must take consideration of length and activation to consider the muscle's function. Now, in my lab, I'm particularly interested in one particular phenomenon called doublet potentiation. Now, this is the increase in muscle force with the addition of a pair of muscles of stimuli at the onset of activation. So what we're looking at to the right here is the force being activated with a low frequency stimuli in the dark black line, and then the addition of a stimulus in this thinner black line. And what you can see is this nonlinear increase in force, so that the force nearly doubles. And then we see that the force is sustained throughout the activation. We know that the nervous system utilizes this pattern of activation during both fatiguing and ballistic contractions to maximize muscle force. And while this phenomenon has been documented for more than 50 years in humans and other animals, the mechanisms underlying doublet potentiation are not well understood. So you might think that calcium underlies this increase in force. And so we can look at calcium concentration, which is this top graph here in the sarcoplasm of a muscle. And indeed, the calcium increases and spikes with the addition of a doublet. But you also see that it drops off really quickly within about 25 milliseconds. So while it might explain the increase in force, it doesn't explain the sustained force that occurs for several hundreds of milliseconds. So cross-bridge models cannot account for the sustained potentiation that we observe. Many researchers have suggested that doublet potentiation could be due to the engagement of an elastic element. And this is consistent with the idea that doublet potentiation increases with stretch and then it decreases with shortening. And so this led us to the hypothesis that the elastic protein titin could be contributing to doublet potentiation. And so just to remind you, titin resides in the sarcomere of a muscle. So we're looking at a sarcomere here to the right from Z line to Z line, and the thin filament here in gray and the thick filament in the middle. And it's this red structure that is titin that spans from Z line to M line. And in the middle here is the spring element of titin. And we know that that part of titin stretches with muscle stretch, and it's responsible for passive force. We also know that titin stiffness increases in the presence of calcium, and it binds to actin at lower calcium concentration than myosin binds to actin. So this led us to the idea that doublet potentiation is the result of titin-actin binding that remains bound at low levels of calcium. So using ex vivo muscle experiments, we can test our hypotheses about doublet potentiation. And we use mice with mutations to specific regions of titin. So in our data today, I'm going to show you data from an N2A mutant. So this has a really tiny deletion in the N2A region of titin, which is associated with binding to actin. Whereas we use a different mutant called a PVK mutant, which has a really large deletion in the PVK region, which we know is responsible for passive force. And we're going to look at doublet potentiation in cyclical movements. So we're going to impose the muscle to sinusoidal length changes. We're going to stimulate the muscle at a low stimulus frequency, and we're going to add a doublet and measure the force and the work with the addition of a doublet. All right. So what we're looking at here are work loops from wild-type mice and N2A mutant mice, and the muscles are being stimulated with and without a doublet stimulus, and they're being activated at a short length and activated during lengthening. And so what we see in the wild-type mice is that there's a greater than 50% increase in net work with the addition of a doublet. And not only do we see an increase in force during lengthening, but we also work during lengthening. We also see that work increases during shortening. But we see a less than 5% increase in net work with the doublet stimulation in the N2A mutant. And this suggests that this small N2A deletion nearly eliminates the doublet effect. And it supports the idea that titin binds to actin in a calcium-dependent manner and contributes to the doublet potentiation. When we look at PVK mutant muscles, we see a different pattern. So again, just to point out, we see the doublet or the wild type muscles here in the top left, and we're looking at the PVK mutant here in pink. And so during active lengthening, we do see doublet potentiation in our PVK mutant, but to a lesser extent than what we see in the wild type muscles. So we see a 30% increase in muscle work during active lengthening, but we see no increase during shortening. If we change the phase of activation and look at when the muscles are being activated during shortening, we see that the wild type mice show a 25% increase in muscle work and nearly no increase in muscle work in the PVK mutant. And so these data show that the PVK deletion reduces the doublet effect. It supports the idea that titin is binding to actin in this calcium-dependent manner, and it suggests that stretch increases the contribution of titin to muscle work. And so hopefully what I've convinced you with these data is that titin is a tunable spring in muscle that can account for doublet potentiation during dynamic movements, and that the N2A region and the PVK region of titin are necessary for doublet potentiation, and that they support the hypothesis that titin stiffness increases with activation and remains elevated long after crossbridge cycling ceases. And so before I leave you today, I just want to remind you of all the benefits of isolated muscle experiments. So these experiments allow us to make predictions about molecular mechanisms and also about muscle function in vivo. And classic isolated muscle experiments allow for control, but are limited in predicting in vivo muscle function. The work loop experiments do a better job of predicting muscle function in vivo using dynamic muscle changes in muscle lengths. And from Nye data, you see that using mouse mutants in vivo, ex vivo experiments can help elucidate the underlying molecular mechanisms. All right. Thank you. I'm going to stop sharing. And it's a little bit long, but maybe we have one time question.
Yeah, thanks so much, Jenna. Yeah, questions for Jenna before we move on?
All right, well, maybe Kiisa, you can tell us about the avatar.
Okay, can everybody see my slides? Yeah. Okay, yeah, well, thanks so much, Jenna, for that great introduction to the work loop technique. And today I'd like to talk a little bit about a variation on the theme. So we're gonna use, let's see what's going on in the chat first. Let's see, there's a couple of
Kiisa, it's just me letting people know they can put questions there if they want.
Oh yeah, yes, good. Thanks, Greg. Yeah, absolutely. Put questions in the chat and we'll also have more time for questions at the end. Yeah, so I want to tell you a little bit about an avatar experiment that we've recently developed. And so what we're trying to do is move ever closer to developing ex vivo muscle experiments that can inform in vivo muscle function. So I'm just going to briefly say that the work I'm going to talk about today was done by a big team and not just me. And I particularly want to call out Caitlin Bemis, who's really advanced the development of the avatar technique in my lab and is now a postdoc in Monica's lab, working on avatar experiments in guinea fowl muscles. So Jenna referred to this gap between in vivo dynamic muscle function and ex vivo muscle experiments. And modeling papers actually sort of emphasize this gap. So we can do in situ work loops in muscles from rats, and we can measure their in vivo behavior. And then we can use various different kinds of models to predict the muscle force under different conditions. And what these data show is that get much better predictions of the muscle force in situ and ex vivo experiments than we do during in vivo locomotion. And actually, as we increase the frequency of the movements of the muscle in vivo in rats from walking to trotting to galloping, we also see that particularly at the high frequencies, the muscle models seem to fail. So can we use muscle work loop experiments to try and better understand in vivo muscle function? And if so, how would we go about doing this? So what we've done is to take the classic work loop experiments, and Jenna really did a really nice job of describing them. So we take a single muscle from a mouse and put it on the arm of a servomotor force lever where we can control the length and measure the force, or control the force and measure the length. But what we do is, instead of using sinusoidal length trajectories, as is typically done, or sawtooth trajectories sometimes in a typical work loop, we actually take length trajectories from in vivo strides from different animals. And then we can begin and end stimulation at the same lengths approximating the in vivo EMG. And then we stimulate the muscle at submaximal voltage, because in vivo, the supermaximal voltages that we can achieve by stimulating muscles in a bath are never, the in vivo forces never approach the maximum isometric force. So we're trying to have the muscles of the mouse experience similar strains and activation as that, for example, a guinea fowl muscle would experience in vivo during running on a treadmill. And so the first example I want to briefly tell you about is going to be the guinea fowl lateral gastrocnemius muscles during running on a treadmill over these obstacles. And these data are from Daly and Winner 2011. So Monica instrumented the lateral gastrocnemius of guinea fowl. So the fascicle lengths are measured using sonomicrometry crystals. The muscle forces are estimated using a tendon buckle. And there's also an EMG signal from the muscles as the birds are running on the treadmill. So we have kind of 3 different types of strides that the birds exhibit. So the first one is when they're running on the level surface of the treadmill, and you can see that the muscle length, the EMG, and the forces are very, very similar from stride to stride. One extension retraction cycle looks very much like the next, and the EMG and forces produced are also relatively similar. When the instrumented leg steps up onto the obstacle, though, we see that we can have higher EMG, higher forces, as well as a perturbed strain trajectory that looks really different from the others. And when the instrumented leg happens to be the one that steps down from the obstacle, what we actually see is we can have very much higher EMG, but it's often accompanied by much lower forces. So we have this natural variation, stride to stride variation in the muscle length, activation, and force. But in the bird, these are all confounded and no 2 strides are identical. And you can kind of see that this is like all of the maybe, 45 strides from a single animal in a single trial on a treadmill. And so you can see that the strides with the red asterisks are when the instrumented legs steps up onto the obstacle, and they can be accompanied by, perfectly average EMG or, much higher EMG, but the forces are always higher regardless of whether or not the EMG is higher. And likewise, we can see in these traces the strides marked with asterisks in blue where the instrumented leg steps down. And interestingly, these strides can also have very high or average EMG, but the force produced by the muscles is always very low regardless of whether the EMG is high or not. So the perturbations have the effect of increasing step-to-step variability, and they reveal patterns in the data that suggest uncoupling between the muscle activation and the force. And so here just now we're looking at many, many trials from many individuals. And here we're actually looking at work loops. So the muscle length is on the x-axis and the muscle force is on the y-axis. And you can see that when there's no obstacle, we get this sort of L-shaped work loop pattern where the muscles lengthen and then they shorten and the force rises associated with foot contact, and then the force decays and we go through the loops again and again, and every force is nearly identical. But you can see that when we have obstacles on the treadmill, we get a lot of different work loop shapes. And here I just selected a few strides. So the red and the magenta are strides when the instrumented leg steps up onto this treadmill. The blue is when there's no obstacle present. And the green is when the instrumented leg steps down. So we can have a large amount of positive work during the steps up and a very small amount of work during the steps down. And it turns out that there's a 40-fold difference in total work between the up to a 40-fold difference between strides stepping up onto the obstacle and strides stepping down. So the question is whether we could replicate this in ex vivo experiments by exposing the muscles to these different strain trajectories with constant activation. And before I get there, I also just want to tell you another interesting aspect of these data. So Siwoo Zhang in my lab analyzed actually the delay between the EMG onset and the muscle force development. And this is in a series of about 350 strides from the same bird. And what you can see is the nominal excitation contraction coupling delay is about 25 milliseconds in these birds. But what we see is that there's a really large variability in the delay between activation onset and force onset in the strides of these birds. And it can be anywhere from around twice the excitation contraction coupling to delay to as much as 5 times longer. So we also need an explanation for why there seems to be no constant EC coupling delay in dynamic movements. Rather, in these dynamic movements, the delay appears to depend on muscle length and velocity. So let's see what we can do by giving ex vivo mouse muscles these strain and activation inputs from the in vivo birds. So I'm just going to monitor the chat. Okay, so Greg asked about scaling, and we'll get to that. Okay,
I think these are questions to address later.
So here we have these different in vivo work loop shapes, and here's just the typical rectangular sinusoidal work loop shapes, and Jenna was showing some of these earlier that you get from a typical sinusoidal work loop experiments. And so why are the shapes different in vivo, and what determines the in vivo forces and work per cycle? And I'm just going to go on to the next slide in the interest of time. I'm trying to watch my time here. Okay, so what we do is we scale these length and inputs shown in the middle here we scale these to try in vivo, and here's the ex vivo data here on the right, to try and match the in vivo rise in passive tension. So that puts us the starting length and also the amplitude of the length change puts us at a relatively similar place on the length tension curve that Jenna was talking about earlier. And then we actually activate the muscles at the same length and for the same duration as in vivo. So we always activate the muscles at the same length for the same duration and with the same amplitude in all of these experiments. And so interestingly, what you can see is that you get a very similar work loop shapes in the ex vivo work loop experiments with the in vivo strains as you do, in the guinea fowl during the in vivo length changes, despite the fact that the activation in all of these starts at the same length and is for the same duration. And here what you see in black is a sinusoidal length trajectory with the same starting length and activation duration. And you can see that it doesn't really replicate any of the in vivo work loops. And so if we look at the next slide, you can also see that there's a very good correspondence really between the ex vivo and in vivo work loop shapes. And there's also a very good correspondence between the delay between when the activation starts versus when the force rises, right? So even with constant activation, we get a pretty good correspondence between the in vivo and the ex vivo work loops. And I just want to point out that we'd get a much better correspondence, and I'll show you some data relating to that in a minute. We get a much better correspondence if we were to match the activation to the actual in vivo activation that was observed in these different strides. So I want to just really briefly give you a second example, but I don't want to take up too much of our time here. But in this example, this is the work of Caitlin Bemis. And so what she did is she actually took in vivo data from rats running on a treadmill while they were actually walking, trotting, and galloping on a treadmill. And she actually improved the technique in a couple of different ways. So she used the matching in vivo EMG duration. And then she also accounted, so in all of these experiments, we account for differences in the excitation contraction coupling delay in the in vivo and ex vivo experiments. And then she actually found that because the in vivo activation is about 80%, or the ex vivo activation is about 80% of the maximum, that she could optimize the stimulation duration, actually shorten it, and get a much better fit to the in vivo force data. So let me just briefly show you her results. So she looked at walking, trotting, and galloping strides from the rats, and she measured the R-squared between the muscle avatar and the actual observed in vivo force data. And so you can see that for all three strides, the sinusoidal length inputs really did a poor job of predicting the in vivo force. And the avatar did a much better job. And these little boxes are the average R-squared from Hill models for the same data. And so you can see the Hill models and the avatar do about the same for walking and trotting, but they do much better for galloping than the Hill model. So there's something that the avatar is catching about how muscles work at high frequencies that the avatar is missing. And I think in the interest of time, I should skip over these really quickly, but this just shows Caitlin optimized stimulation protocol where she actually shortens the duration of stimulation, and that greatly improves the fit. So Greg, to answer your question, we actually scale the activation based on the rate of the passive force rise, and then we scale the stimulation by shortening the delay to account for the fact that the muscle force decreases much slower in the avatar than it does in vivo. And the same for trot and gallop. So I want to just wrap up, and I'll stop sharing my slides in a minute and answer other questions, to say that there's a lot of strengths, many of which Jenna already mentioned, to being able to do these experiments ex vivo. So we can precisely control the length and stimulation, which vary continuously in vivo, and no two strides are the same. We can, because we can, for example, do the experiment where we can have the stimulation always occur at the same length and for the same duration, we can reveal the importance of strain and strain rate in determining muscle force output. And we get a surprisingly good prediction despite all the differences in muscle species stimulation between the in vivo and the ex vivo case. But of course, there are limitations, and two main ones, but by no means an exhaustive list, is that the in vivo activation and ex vivo stimulation differ in important ways, right? And we need to understand this better, what the consequences are for modeling the in vivo muscle forces. And also we need to validate or evaluate the avatar technique using muscles of different sizes, fiber types, and architectures from different species. So I'm going to stop there, and I'd be happy to answer any questions. So Greg, did I answer your question about how we scale?
Yeah, I think that's a great way to approach it, is to, get the force patterns to more closely match the in vivo experiments by tuning the activation in the avatar preps. I think that's great. I wonder if there's like a fundamental sort of time, could it be derived how to do that without fitting, is something I've been thinking about. Like, how does the scaling of activation, is there math behind it that predicts what you had to do in order to make the activation scale well to get the right forces? I don't know. It's a bigger picture question that we don't need to answer.
Yeah.
It's something I've been thinking about.
I'll take a quick shot at answering that, and then I see a raised hand as well. So So we're working on like system. So there's always been this question that's never really been answered. And many of us, including Madhu, really want to know what kind of a material is muscle. And, you know, if we really could say, oh, muscle is, you know, a viscoelastic solid or a viscoelastic fluid or a, you know, weight material with a spring in series, then we could, you know, then I think we would have the math to do that. So I think that the way forward to make it more theoretical and have a mathematical basis is to develop predictive models of muscle that we can then, so wouldn't it be cool if we had a muscle model, we could apply it to the in vivo guinea fowl data and to the ex vivo data, and we could, you know, we could say, you know, are the materials the same? Are the material properties the same? How do the material properties differ? How do the time, you know, how do the, you know, the lags due to the viscous behavior of the system differ between the two preps? So I think that that's where I am trying to go with that, but it's a big, it's not a simple problem. So, okay, so BKN, do you want to unmute and ask your question?
Sure. Can you hear me okay?
Oh, yes.
Yeah, so, you know, often when I think about both muscle and limb mechanics during locomotion, I think about them in a phase-specific manner. So, for instance, what are the roles of specific muscles during weight acceptance versus propulsion? But with work loop experiments, you're sort of considering the full gait cycle at once. And so I'm having a little bit of trouble. I'm now realizing what has confused me about work loop experiments is that mismatch between how we think about gait in a phase-specific role of muscles, where muscle might absorb energy during weight acceptance, but then perform positive work at push off. So when doing work loop experiments, do people typically try to separate those phases to talk about the different roles of muscles throughout the cycle, as opposed to just looking at the cycle as a whole?
That is a really great question. And I think it really shows how, you know, we all have different perspectives and are interested in different things. So what Jenna showed about you know, if you stimulated different phases, like a muscle, if you stimulate during lengthening, you can, you know, then the muscle acts more like a brake. If you stimulate during shortening, it acts more like a motor. So during lengthening, it does negative work. During shortening, it does positive work. And when you stare at the work loops from a particular muscle enough, you can kind of see the phases of the gait cycle in there. So you can read from the strain, you know, sort of what's happening. So lengthening is passive protraction of the limb, and shortening is active retraction. And the interesting thing about the guinea fowl muscles is that they are strictly active only during shortening. So they're really doing positive work. But the strain trajectory alone, and what the mouse experiments are saying, is that the strain trajectory alone can really regulate over, you know, a factor of 40-fold, you know, just even during sort of the shortening and weight acceptance phase. So the end of swing and into the beginning of stance can really, which is when most muscles are producing force during locomotion, at least most muscles that are contributing to the positive work, you know, that the strain trajectories, and particularly transients associated with foot contact, can really modulate how much work is done by the muscles, even with constant activation. Did that answer your question?
I just wonder if there's sort of quantitatively, if you were to take the work loop data and analyze that data, you know, just over specific subphases of the gait cycle, is there a way to really tease apart that information? But yeah, it's just something that has now become a question as you all were presenting this morning, but you pass it along to Monica's group.
Okay. Yeah. Do you have a?
I think Katie wanted to say something, but before I pass it over, I'll just say, yes, we could absolutely do that with work loops, though I don't think the classic work loop approaches have typically done it that way, but we could look at it that way more. But Katie wanted to add something.
Oh, yeah, I was just going to answer Greg's question more directly, like with numbers, that like if that's okay, Kiisa. Okay, but like, so in the mouse EDL, when we use the rat medial gastrocnemius strains, for walk and trot, it was about 50% reduction, but during gallop, it was about 25%. Where it gets a little bit more complicated is when we use, which data we didn't present here, use the guinea fowl strain and EMG activations for both the lateral gastrocnemius and the digital flexor. No optimization was necessary, and it actually performed better than optimized numbers in the rat medial gastrocnemius. So it gets a little weird there, and I'm not sure that we've totally sussed out why two closely related species needed so much optimization when the guinea fowl did not. So.
Very cool. Thanks for adding to the original question.
Manny.
Hi, Kiisa. I'm just trying to get my head around the difference that exists in vivo in terms of the relationship between EMG and the force decay or relaxation rate, and the one in situ or ex vivo. We do have these fancier stimulators where we can, in real time, change both rate coding and voltages dynamically. And so I'm trying to figure out what is it that's fundamentally different about the stimulation protocols versus how the nervous system interacts with skeletal muscle. And is there a way for us to sort of push that in a way to make it more realistic in our isolated muscle experiments? So the difference really is pointing out something fundamental, right? It's saying your stimulation protocols are not really the same. And so I wanted to get closer. And how do we sort of get there?
I think that that's a really great question. Hang on just a second. I think that that's a really great question. And we do want to make them better. So I think, we need to incorporate, to ask the question. So Siwoo Jong did do some avatar experiments where he used, he modulated he didn't just use, constant amplitude square waves, but he matched the amplitude of the square waves to and he found that it didn't improve the fit at all. Now that's just one case. So we need to keep trying that and seeing if we can make it better. I do think that you can get like 90%, as Caitlin's, showed in the rat, by just reducing the duration of the stimulation. And Monica says, watch the time, and we'll switch over to Greg just after this question. But I think it's a really important question. So you can get, and the idea behind that is that, I think the fundamental limitation of the ex vivo stimulation is that we can't get to very low percentages we can get nowhere close to what the real percent of maximum isometric forces in vivo. So these experiments are all done at 80% of the super maximal in vivo force. And when you have more force, it just takes longer to decay, right? So I think we need to absolutely explore this question, and we need to nail it. But I'm hopeful that the different, it boils down to calcium and the rates of calcium release and reuptake and the effect that the calcium has on the, changing the structure of the proteins within the sarcomere. And so I'm hopeful that it might not be overly complicated. So yes, there's a difference, but it isn't big, and you can get quite good results by just tuning one parameter, which is the duration of the activation. Okay, so I'm going to hand over the screen and the mic to Greg.
Okay, can you see slides and hear me? Thumbs up. Okay. So I'm gonna push us a little bit further beyond Avatar, hopefully, in this next section, and think about how we integrate closed loop dynamics between muscle and a load, say a body of an animal. I'm not advancing. There we go. So I just want to first motivate this by reminding us all that real world locomotion is complicated, right? So the schematic I have here is something like a classical work loop. We've got stimulation going into a muscle, and then we know that length and velocity dictate or help dictate what the force is coming out. But of course, in a real animal, there's a body that those forces load or drive. And then there's a body state. And in between the muscle and the body, there are tendons that modify the length and velocity that the muscle sees. And then there's a brain and a spinal cord. And of course, there's also sensory feedback, right? So when you look at a human or any other animal move in the real world, all of these cascading feedback loops are at play. And the approaches we've looked at so far are really only snapshots within this. So what we've been trying to do in my group and in others is sort of build from bottom up on top of a classical work loop to get block diagram behavior sort of on the benchtop that looks more like this. I just want to remind us all, and some of this has been covered already, but I think it's good to go over. What are some of the approaches that people have used to do this? Well, straight up in vivo experiments are great, especially when you can instrument individual muscles. The classic example of that is work by Monica and her group and others who have done these experiments in animals, including humans, when you bring ultrasound and other tools that are non-invasive to bear. Pro is that you've got an animal that's behaving stably in its natural environment. Cons are that you can't really tell what any individual muscle is doing. You don't have a lot of control over modifying around the natural behavior, things like activation dynamics and length changes. On the other hand, if you go to in vitro experiments, you can, as we've heard already nicely from Jenna and Kiisa, you can really isolate the dynamics of a single muscle or maybe even a single muscle tendon unit, but you don't have real movements, right? You're not coupled in an emergent way to, say, body dynamics, mechanics, sensory feedback's not at play, et cetera, et cetera. So another approach is modeling and simulation, which would be great if we had good models of muscle force. So that's the key problem there, is that pill-type muscle models miss the boat, as Kiisa said nicely, especially for high frequency, highly transient and unsteady movements. So the avatar approach is starting to fill this gap between in vivo and I feel, and we already heard about that from Kiisa and her group, where we can start to get more realistic length change and activation patterns on the benchtop by stealing them from recordings in naturally moving animals and playing them back in our experiments. So from here on, I'm going to talk about another approach that builds on top of the classical work loop, which is, it's really outlined, I think, pretty nicely in this review paper that I worked on with Emily Abbott and Simon Spomberg. So I just want to plug that here. If you want to read more details about any of the stuff I'm talking about here today, go check out that paper. And really the idea here is, okay, you've got this complicated network of feedback loops. You can peel it away to the classical work loop, which looks just like that, where all of these things are controlled on the benchtop. And then you can start adding complexity to try to build up to that full closed loop system that I had up there just before. And the classical work loops, I'm not going to get into. You've already heard from Jenna how those work. You can do things also like impose strain patterns again that are more naturalistic, like Kiisa talked about. But one key thing that our group's been interested in is how the series elastic tissues that we all know they're between every muscle and the skeleton, but especially in distal muscles, like the ankle muscles in any species, really, it's a prominent structure that we need to take into account. So that would look something like this: adding the tendon or leaving the tendon on a prep in a work loop preparation. And with that, a little bit more detail on how that looks. And this is work going all the way back to my postdoc days when I was working with Manny and Tom's lab, and then my student Ben Robertson working in our own lab kind of extended some of these approaches. But the key thing here is instrumentation, that if you take sonomicrometry techniques and you layer them into the classical work loop, what it allows you to do is separate the motions of the muscle compartment and the elastic tissue compartment. And it gives you an idea of how things like phase, turning the activation on earlier or later in a cycle, play through the series elasticity to get muscle dynamics. And so that's really what we did here, and verified that in the center panel down here, this looks a lot like what you would record in steady running, for example, in a turkey or a human, where actually, even though the whole muscle tendon unit's going through a large sinusoidal amplitude, the muscle itself is behaving very much strut-like, kind of like Jenna presented early on, where you see we can recover this strut-like behavior because the decoupling between the muscle and the muscle tendon unit is captured by the phase that we turn the muscle on with respect to the stretch of the tendon. Okay, so great, we've got tendons built in. What's next? Well, let's try to, instead of impose the length changes directly on the muscle tendon unit, it might be good to play the forces that are generated through a body and let mass and inertial and gravitational dynamics dictate the length changes. So the analogy here would be like vertical hopping in place driven by an ankle plantar flexor, where you stimulate the muscle, force plays through the muscle tendon unit, drives the body. The body, in this case, this kind of speaks a little bit to James's question. We can generate a virtual aerial phase if we prescribe the dynamics through a model that incorporates that. It's sort of what we've done in my lab. These preps don't move forward. There's not an actual physical body, but we can use modeling and simulation. Our approach has been to model the body dynamics. Again, Ben is popping up here. Other groups that we were inspired by who came before us that were coupling work loop experiments to real world loads, put them into physical interfaces that had real world dynamics, especially in swimming, the work by Chris Richards. And then even after that, others have kind of revisited the fluid dynamic interaction with work loops by using modeling to get at the fluid dynamics as well. And that's some work that was happening in Lena Ting labs quite a while ago that we've also been really inspired by. So what does this let us do though? It starts to let us understand how force production in the muscle tendon unit lead to emergent behaviors of the body. And what that looks like is something like this. So this is time on the bottom. I have force and length of a muscle tendon unit. So the tendon's in play here. The red part right here is the muscle compartment, and the rest of it's the whole muscle tendon unit. So what we do is all these now start with stimulation. We're not prescribing the length. We just start stimulating the muscle feed forward, produces a force. The force drives a virtual body and things happen. And what you can see here is like some really cool things, like the muscle has to accelerate up to steady state. So you get some transient behavior emerging before the steady cycle begins. And then the steady cycle you fall into is really now driven by phase and not time. Again, kind of speaking a little bit to James's ideas about how do we square the circle of time versus phase and work loops versus real animal movement. Okay, so just to go through this a little bit more quickly, not quickly, but a little bit more carefully. Take a muscle out of an animal. In this case, it's a muscle tendon unit from a bullfrog. Attach it to a servo motor. But the trick here is to make the servo motor feel like a body by putting a model in software that has mass and inertial dynamics. Then we stimulate things to get it going. And what happens is, we get this happening, right? So you get a hopping on the benchtop type situation. Okay, so that's basically the crux of it. That's how you bring real world loads in. How do you get some more of the detail of that complicated cascade of feedback loops to work? Well, on the sensory side, we've been trying to use different preparations where we can record from the spinal cord. This is work led by Emily Abbott with some help from Laksh Punuth and Amral Al-Sharif and Shu, who are bringing it forward. But the idea here is do exactly what I just showed you, but also make recordings from the sensory side of the house to try to understand how these closed loop behaviors that now involve sensory feedback might be. Other things you can do are add like exoskeletons or other external environments into your coupled model in software, and we've done some of that too. And the other thing that we're starting to try to do now is simulate changes, say, in the substrate height. So can you do the falling in the hole experiment like Monica's done in a real animal, or we've done some of that in humans too, but now on the benchtop? And the answer is yes. All you got to do is change the virtual ground height in your mathematical model. So the ground's not there, and the prep will stretch further before it experiences the dynamics of ground. And that lets you sort of understand what might happen, what emerges in terms of substrate heights up, in this case, where the muscle now automatically does some positive work just because of kind of phasing that's built into the physics. And then if you drop the system down into larger and larger virtual holes, emergent negative work, it comes out of the system too, which is quite nice. None of this involves sort of any shift in the phase of the feed forward signal, and it doesn't involve any sensory feedback. So there's a nice coupling here between physics and muscle dynamics that emerges from these preps. Okay, so we've gotten all the way back to sort of this, bottom up from classical work loops to open loop sensory feedback on the bench. The hard part is going to be to integrate spinal cords, and we don't know how to do that yet, except for to observe humans, which is what we're trying to do sort of as part of the workshop series later in the summer. We'll show you some ideas we have about how to bring these work loop preps sort of into humans, where we can measure all the stuff I just talked about using like ultrasound, EMG, highly instrumented pedal dynamometer. And then we know that human's closed loop, right? They have sensory feedback and feed forward stuff going on at the same time. Okay. I think I'm right at time. Maybe people can hang around longer. I have till 1:30 if people want to chill. But here's the QR code for people to give feedback, and maybe I'll stop sharing, and hopefully there's time for people to hang out, and we can get some more Q&A going. Okay.
Maybe we can put the QR code up for just a little bit longer in case people want it, but it would be great to give time for more discussion.
And also the QR code will be in the slides on the CMSI website. So if you didn't have time and you want to do it later, you should be able to get the feedback just by going to the grid.
We'll post it in the Slack.
Can you guys still see the QR code now?
Yes.
Yes.
Okay, good. Great.
Yeah, that's a great idea.
Probably posted good now. Yeah, we can probably stop sharing now. That's cool. Thanks.
And Alice already put it in. She put it in the chat as well, and I'm sure she'll be putting it in the Slack.
Did I stop sharing? I think I did.
No.
Or
No,
you guys are still seeing it. Oh gosh.
Yeah.
I'm working on it.
Well, if anybody wants to think of questions while we're working on that, you can put them in the chat or go ahead and raise your hand. Or just unmute and talk. Manny, do you want to just unmute and ask your question?
Sure, I can talk. I just wanted to add another utility of these types of experiments that particularly the ones that Greg mentioned with real world feedback. I think they can actually really serve as a nice way of testing the conditions under which muscle models perform well and the conditions under which muscle models perform poorly, because you effectively have a highly controlled model that a real muscle can act against. You can run that same model being actuated with a hill type muscle model or a rheological model or any type of model that can take in inputs. And it really starts to show you, okay, what are the conditions where these models start to perform poorly? And that in and of itself really starts to point at places where our understanding isn't quite as good as it needs to be in terms of our understanding of skeletal muscle.
Yeah, Manny, you're dead on there. I think part of what we're trying to do, especially in these unsteady situations where we create virtual holes, is we drive the recorded data through a fitted hill type model. And we're able to show exactly where it's bad, which is right on impact when the muscle's when activation is coming up and the muscle's being rapidly stretched. And we know a lot about this already from like Kiisa's work and others on the benchtop that have shown that those are the really bad spots for hill. But our data suggests exactly the same thing, that it's in rapid stretching and transient, especially back to James's idea, in the transitions from one type of mechanics to another, like coming out of the air and onto the ground. And that's, I think, where we want like intrinsic, I'll use the word intrinsic. I know there's some debate about what the right words are here, but where like Titan and cross bridges can do their thing inside of reflex latencies.
Neuromechanics lab.
All of us here. Actually, it's me, Kristen Jakubowski. I have a question. It seems like each lab and each group sort of has a muscle that they like or a species that they like. So like Greg was presenting work on the plantaris, but each of these mussels has very different architecture, very different passive mechanical properties. And as Caitlin pointed out, like the scaling and sort of the optimization can vary quite a bit between mussels and across species. So do we have a sense sort of how these properties really influence the results? Like do the results that Greg presented in the plantaris, do they work also in the algae or in other mussels? And how can we start bridging those differences across scales and across species?
I can say one initial thing in response to that question. I mean, I think a lot of it boils down, if you consider the whole muscle tendon unit, architecture matters, right? So like applying the same muscle tendon unit or joint motion to a muscle that has a lot of elasticity and maybe even interesting pennation, etc., will give you much different force output without even changing the activation. So we can't forget that the structures around the muscle, and there are lots of them. We're learning more and more, especially also about 3D shape change. We have a lot of people in this group, like Sylvia Blemker, that are revealing that complicated changes in the geometry also matter. And that might be that's a difficult thing to model between species potentially too. We need better ways to do that.
I'll maybe say one thing and see if you guys agree. You're free to disagree with me. So if we were God and we knew what kind of a material mussels are, it seems to me that this strain trajectory, so with the theory of materials, that this strain trajectory itself is a reflection of the sort of the long axis deformation response of muscles to all the applied forces, like by the tendon, by the antagonistic muscles, by ground reaction forces, etc. Right? And so I think some of the answer to this question is, to Kristen's question, is, doing the exact same experiments with the exact same, in different muscles. And, and I guess like, where I'm kind of at in my thinking is I think like having a good muscle model or a good avatar, like, we can kind of get like, 80% of the way there, sometimes a lot closer if, by chance the muscles are similar, just by assuming all muscles are identical, but obviously they're not. And obviously I think muscle size matters, and everybody's, on the bandwagon that muscle mass matters. And so, yeah, I think that we can, deploy the comparative method to work out some of these things. And, one thing Monica and I are always talking about is like, we need an army of young, ambitious scientists. We need to train an army of young, ambitious scientists because there's a lot of work to be done to get to the answer of that question.
Katie wants to add something.
Yeah, and I think a perfect segue off of Kiisa. So what we did for one of my chapters is we took the same rat medial gastrocnemius strains that we saw, and we actually inputted them into mouse medial gastrocnemius to see the differences of how much the fascicle length changes actually can tell us about a whole muscle tendon unit of a similar, assuming a similar structure even across species. And there were some interesting results there. Obviously there was less need for optimization of stimulation, but it does, I don't know, I don't want to give it all, we don't need to, that's a whole nother topic, but we're trying, right? So we're trying to take data that we've already seen and put it into other muscles and other species to see how it will react.
So I have a follow-up question, and I'm gonna ask you to maybe air your dirty laundry.
Okay, we can.
Are the muscle choices that have been made based on convenience and accessibility? Because I think we're having trouble generalizing a lot of measurements to other muscles because the tendon's not long enough, or the surface, it's not near the surface, so we can't measure it, or for instance, like with ultrasound, the motions that we want to see, and like it might be in a hip muscle that it's really hard to attach. But I know, like in animal experiments too, there are reasons that we love studying the triceps surae because they have a beautiful tendon and you can attach it to things. And then when you start to get at other muscles, like let's say vastus medialis, like it's deep, it doesn't have a clear connection point, and like you just can't even do the experiments on them. Or in some cases, the architecture means that it lasts longer, or so there's all sorts of other details. So tell us why you picked your favorite muscle.
Who wants to start? That's absolutely one of the big challenges, right? We study the gastroc because it's readily accessible, but then also so many people have studied it that we have a big body of knowledge to help work from as well. But maybe we could also think about ways to use these existing systems and manipulate them in a way, like we can do things like maybe systematically remove connective tissue and test the muscle with its full in series like tendon, and then reduce it, test it again, and see how some of these factors influence. And that could get us a little bit closer to replicating different kinds of muscles. But it's absolutely also the case that we don't, it's kind of a scattershot approach because we don't have the ability to measure all of the muscles in the system, and in particular the ones that are proximal and don't have free tendons. We just don't have an easy way to put them in a rig. Anybody else can feel free to chime in if you want to talk about your species.
I mean,
Go ahead.
I'll throw it out there. Like, if you have a special muscle or your favorite muscle that you want to teach me how to do the preparation for, like, I will go there and learn it, and I will do our experiments in that muscle. And then maybe we can answer that question more directly, but that's my pitch for everybody. So,
but that's absolutely what the spirit of IMSI is, that we should be having these conversations and coordinating with each other to say, where are the big gaps, and then try to really systematically tackle them together.
Yeah, no, I agree, because I ran into this problem when I went into wanting to collect from, let's do every muscle. And then as a young scientist, the advisor's like, well, we can try. And then we try to do, instead of gastroc and EDL or TA or something, and then just with our rig, I literally just cut off the blood supply to the leg.
Yeah.
It was like experimentally very, very difficult.
Yeah, there are just some muscles you can't get a prep going, but I think Manny wanted to say something too.
Well, I think I've selected muscles in sometimes largely because their activation level in vivo was closer to maximum. And for me, the biggest hole is submaximal recruitment. So Kiisa brought up the God idea of like, if I was God, where I would want to look is at the interface in vivo of an active and passive fiber right next to each other. What does that interface look like? What is the mechanical interaction between tissue that is active versus tissue, being immersed right next to tissue that is passive, particularly given that most behaviors, the muscle is going to be 80% passive. What are those fibers doing, and how are the active fibers interacting with those? That to me is a fundamental thing that we're still missing in our isolated muscle experiments, is that understanding of what it means to be a passenger on a submaximally activated muscle.
So Manny, like what about like Hugh Moss's stuff where he brings lateral muscles that are laterally located along for the ride? Is that in work with experiments? I think he has some work along those lines. Is that what you're getting at?
I mean, even more complicated, really. So that's a simplified version where you've got a fully active muscle next to a passive muscle, and you're sort of looking at the interface between them. I'm talking about at the single cell level, you have cells that are active surrounded by cells that are passive. What do those passive cells look like, and how is that active fiber interacting with the passive ones around it? particularly if we're operating at 25% activation most of our lives all the time. That says 75% of those fibers are passive. And the fact that I don't have a good sense of what those look like, is there more tone in vivo that where they're stiffening up to some degree? Is it enough for connective tissue structures around the active fiber to simply act on them in a way that allows for transmission of forces through them? To me, that's the window I would want to look in. Hoops experiments are good in that they're looking at the interface, which I think is important, and they're looking at it in a place where you can see it. The questions I'm asking are really unapproachable experimentally because they just are.
What
So
about
Manny,
robo-physical models?
and I was also going to suggest, the kinds of experiments that Natalie does, where the difference between frequency modulation of force and voltage modulation of force, where so you can achieve the same force by having the whole muscle activated by some small fraction of the total activation versus frequency modulation where you only have some fibers that are activated. And so would that you'd expect differences between those if it matters.
Yeah, I mean, she did some of that work in my lab, and we did find something interesting, which is that the length tension curve shifts way to the right as you lower activation level. And our explanation was that helps those passive fibers that are going along with everything have a little more integrity to be able to transmit forces more effectively. But that's all we could say, that we think that's what's happening, but I wasn't able to think of a more conclusive experiment, and yeah.
Anybody else want to air their dirty laundry about why they pick their particular muscles to study?
I mean, for us, it's pure convenience. Like, engineers who don't really feel that great about doing surgery pick easy surgeries and preps that help answer very specific questions about series elasticity. So we wanted to be sure we had muscles that had large, long tendons. And then, like, on the human side of our lab, what Lena says is dead on. Like, we're totally lost when we go proximal to the calf muscles. Not totally, but like vasti are okay in an isolated situation, but when you move to walking, the probe size creates complications. And if you go even further proximally, you can't fit a single muscle in a single probe, and you have to do these complicated stitching things, and it gets just horrible.
So we're really limited by the tools we have available to measure these things directly across all of the different preps that we're using ultimately, right?
Yeah, the other thing too that I would add about the human side in terms of the calf muscle is that I also find it interesting the groups that solely focus on the soleus versus those that focus on the medial gastroc. Everyone usually ignores the lateral gastroc. So that one is very understudied. But I find that groups either study one or the other and are very hard in one camp or the other, and they're two very different muscles that behave very differently, and there are pros and cons to each of them. But I think it sort of falls within this, that even on the human side, we're still not great at diversity in the muscles that we're exploring.
Well, it's interesting that even, two muscles that you would think should be as similar as they get, like the medial and lateral gastroc. We focus only on the lateral gastroc in the guinea fowl because the medial gastroc has a more complex architecture that we don't really want to deal with. So we just try to ignore it as much as possible.
And it's the opposite in rats, where the lateral gastroc actually more covered up by the biceps femoris and has fibers that are inserting onto the lateral gastroc. So the medial gastroc's actually more convenient in rats, for example.
Mm-hmm.
And I was just going to say that both Caitlin and Nicole in my lab tried using the mouse soleus as an avatar. I mean, the mouse soleus, it's the easiest muscle to take out of a mouse, and the EDL is like quite a bit more difficult. Like it's sitting in a blind pocket and you have to make a blind cut. And so some percentage of muscles just aren't even usable by the time you get them out. But the soleus is a terrible, avatar, at least for the muscles that we've tried, and neither Katie nor Nicole could make the soleus function as a good avatar. And I will say, like, from the point of view of the titin protein, the soleus is an outlier, and it's different from all the other skeletal muscles. So, and it's also sort of uniquely slow amongst certainly the triceps and most human lower extremity muscles that are involved in locomotion. So, but, understanding why, why is it, I think is really, really important. And one thing about the soleus is that it has really low passive tension. So when you're matching the passive tension, you have to, if that's what you're doing, you have to be at much longer muscle lengths. And then, yeah, anyway, interesting conversation.
I have a question for everybody, especially the panelists. If you could grow a muscle, there are people like Ritu Raman. I'm following her work very closely these days. Amazing stuff, where regenerative engineering and genetics approaches to growing muscle in a petri dish offer opportunities that are different and distinct from genetic mutations, for example. What would be your dream prep? Like if you were to collaborate with Ritu Raman, what would you ask her to grow for you? What kind of different muscle that doesn't exist in biology that could branch off of biology if forced would you ask a person who can do that to grow for you so that you could do tests on it? That's probably going to be the final question, I think.
Kiisa, are you going to tackle that question? You're muted, I think.
Obviously, I need to, do more research into these papers. And, I was just going to turn that question back on you, Greg, and say, what kind of muscle would you want? One of the things that this is the way I'll answer the question. And Jenna showed we're really looking at, mouse mutations and transgenic animals, and the people are playing around a lot with the titin protein and making some real, that LPVK mutant that Jenna showed is it's a big deletion. It's like 1,500 amino acids from the protein. And so kind of a different way to look at that is, to use these, naturally occurring and artificial mutants in mice to try and get a feeling for, what are the players doing, but there's a lot of problems with that. So I don't know whether you can use these in a dish preps. I think it would be a strong inference thing to combine those with, sort of genetically altered proteins where you could really, you'd have another knob to turn in those experiments.
Yeah, I think for me, it would be like direct control over the quasi-static properties. Like not in a model, but engineer muscles that are from the same tissue, same size, same geometry, but have very different force velocity curves as measured, and compare them head to head. That would be pretty cool.
But then you could also do the experiment where you have identical muscle fiber type properties and yet alter something systematically about the geometry and the series elasticity, which could also be really cool.
Yeah, agreed.
As far as I know, they're really not getting the connective tissue structure that dialed in either the regenerative side, or there's this sort of almost like a 3D printing muscle that folks are working on. Those have almost no passive tension. The connective tissue structure is largely missing. So I would just encourage them to get better at meeting all of the mechanical requirements that are in place for real skeletal muscles to function properly, rather than so I would say baby steps. I would like to see a muscle that has some reasonable passive tension on it before having grandiose desires about more extravagant designs.
Absolutely. They're still a long way off from replicating a intact muscle tendon organ, right? Yeah. Are we ready to wrap up? Maybe?
3 more minutes for questions. If anybody has one more thing they'd like to share.
And the questions and responses that were added on chat, whoever is the host, can you post that in the Google Drive folder? I think that there are exchanges there that are helpful.
Yeah. I just want to mention too that a lot of the chat questions are about multiple muscles in multi-jointed systems, which I think is incredibly important, right? So how do you couple muscles together in work loop type preparations is an interesting question that Manny kind of alluded to it already a little bit, like muscles that are adjacent to each other, but also muscles that exist together within the same limb at different joints that can interact with each other dynamically.
And I think that's just going to say, do you know whether the chats are recorded as part of the recording?
As far as I know, they're not, unless they're in the transcript. I don't know how. So I might need someone to help me with that. Sorry, but who was speaking?
I'm screenshotting the chat right now.
Whoever is the host,
thanks,
when you
Alice.
close the chat, you will get a text file that's downloaded onto your computer with the complete chat.
Okay, I'll try and. I'll
you have it set up for that.
if I get that, if I have the opportunity, I'll do that. But otherwise, Alice is making a transcript with screenshots.
Okay, so we won't. Yeah, we'll try to.
I just really want to thank everybody. I thought that this was a really great discussion, a lot of participation, and thank you so much for joining. And I also just want to remind you to use the QR code and give us your feedback so that if we do this again, we can make it better. And I'd also want to say that on Thursday at 9 a.m. Pacific, noon Eastern, our next workshop will be by Bert Tanner, and he's going to be talking about computational modeling in muscles. So I hope to see you all there. And I'll definitely put a reminder along with a copy of the link into the general Slack channel